The main feature of eukaryotic cell are their membrane-bound organelles and overall the nucleus that contains and controls the DNA. The nucleus is the far more complex structure into the cell and can be considered its very headquarters that coordinates all the instruction to keep the whole organism alive and fully efficient. In here, the DNA is tightly packed into chromatin. Within the nucleus, we can distinguish a rounded dense area, the nucleolus. It doesn't have any membrane, but is surrounded by dense chromatin. The nuclear DNA is organized into chromosomes, and both replication and transcription take place into the nucleus. The genetic material holds all the information for proteins production. The first step in this process is the transcription, when the DNA is copied into the equivalent version of RNA. The second step is the translation, where, thanks to ribosomes, can be generated the sequence of amino acids forming a protein. The genetic information kept into the nucleus is protected by two double membranes, and through them the nuclear pores select, modulate and controls molecules' traffic across membranes. Nuclear membranes are directly connected to the endoplasmatic reticulum. The endoplasmatic reticulum is a system of organelles made up of two subunits, the rough and the smooth. They further process the new proteins. Their membranes are folded several times, forming sacs, known as cisternae, and tubular structures. They are also involved in the synthesis of certain lipids and cholesterol. The rough endoplasmatic reticulum gets the name from its own aspect, due to the presence on its surface of numerous ribosomes. The smooth endoplasmatic reticulum instead lacks ribosomes. It has tubular structures. Because their complex structure, proteins, after have been processed by the endoplasmatic reticulum, still need further modification into the Golgi apparatus. From here, they are also packed into vesicles and sent to their destination inside or outside the cell. The Golgi apparatus is made up of a series of flattered membranes, forming compartments known as cisternae. A constant flow of transport vesicles from the endoplasmatic reticulum move through these compartments, from the entrance area, the cis Golgi, to the exit, the trans Golgi. Mitochondria generate most of the chemical energy needed to power the cell's biochemical reactions. There are an inner and outer membrane, and between them the intermembrane space, then the cristae and the matrix. The cristae expand the inner membrane surface, enhancing the ability to store the chemical energy into the ATP. And now, let's give a look to the lysosomes. These organelles contain hydrolytic enzymes to break down many kinds of biomolecules. They are involved in various cell processes, including secretion, plasma membrane repair, apoptosis, cell signaling, and energy metabolism. Peroxisomes are small oxidative organelles with a fine granular matrix surrounded by a single membrane. In here, molecular oxygen is converted into hydrogen peroxide, from which peroxisome gets its name. They also perform key roles in lipid metabolism. Vacuoles are membrane-bound organelles. They may contain solids to store nutrients or waste products, or they can be filled with water containing inorganic and organic molecules. Cytoplasm is the name of all the remaining material within the cell that actually is enclosed by the cell membrane. It's made up mainly of water. The cytoskeleton is a dynamic network of protein filaments. We can distinguish three different components: microtubules made of tubulin, intermediate filaments, and microfilaments made up of actin. Their functions are to give the cell its shape and mechanical resistance. They also organize the movement of vesicles and organelles within the cell. The organizing center of cytoskeleton is the centrosome. It contains two centrioles that, during the cell division, migrate to the opposite side of the cell, forming the mitotic spindle. The cell membrane, also known as plasma membrane or cytoplasmic membrane, protects the cell and separate the interior from the outside environment. The cell membrane consists of a lipid bilayer made up of phospholipids. These molecules have a hydrophilic head containing a phosphate group and two hydrophobic tails derived from fatty acids. The membrane also contains proteins and other lipid components. They have some degree of freedom in their lateral movement on the membrane surface, and for this reason it is usually referred as fluid mosaic model. There are also present channels that connect the two sides of the membrane, allowing the transport of molecules inside or outside the cell. On the surface, many receptors make possible for the cell sensing the environment and responding accordingly. A flagellum is a lash-like appendage that protrudes from the cell body of certain eukaryotic cell. Its primary function is locomotion. Within the flagellum, there are 9 fused pairs of microtubule doublets surrounding two central single microtubules. This so-called 9+2 structure is characteristic of the flagellum core, and it also known as axoneme. The basal body is the base of the flagellum. Its inner structure is made up of 9 triplets of microtubules without central pair.